I had a student ask me a question in class the other day that made me think some more about mitochondria. And it made me think about a calculation that I did when I was in graduate school at the Indiana University School of Medicine in a class in the microbiology department at the time with Randy Rosenthal and Rich Hawk. And so I just wanted to go through and do some estimates based on some things with mitochondria to sort of give some perspective about their size and their relationship to other parts of the cell, right? So a typical mitochondrion is, right, we know it's, an elliptical, right? It's an ellipsoid with a folded inner membrane to fit in there. Probably shouldn't have crossed those, but that's all right. A folded inner membrane. And mitochondria vary in size, but I'm going to use an estimate of about a length of about 1.5 micrometers and a width of about half that, about 0.75 micrometers. That would not micromolar, micrometers, right? And so if we look at this, the volume of an ellipsoid, right? If we look at something that is elliptical but round in the other direction, right? So you've got from center out as a distance, from center up as a distance, and then from center to the edge as a distance, right? So you've got distances, A, B, and C. And so the formula for finding the volume of an ellipsoid is that the volume is equal to 4/3 times pi times A times B times C. And if A, B, and C are the same, then you've got a sphere, and you get the spherical volume of 4/3 pi times that distance cubed, right? So if we take those distances and note, right, it's from a center, right, then the volume of our mitochondrion is going to be equal to 4/3 times pi times 0.375 micrometers, right? Half of that distance across. And that's two of our distances, so that's going to be squared times 0.75 micrometers, right? Half of the distance, right, from center to the edge, right? And so you end up getting a volume of about 0.4418 cubic micrometers. And, right, this is an estimate, so more than one significant figure is an exaggeration, but we'll go with it, okay? Okay, so we found that the volume of our mitochondrion is approximately 0.4418 micrometers, and again, cubic micrometers, right? That is an estimate because, as I said, mitochondria vary in size. You see dimensions like I gave, you see smaller dimensions, and so they can vary. So let's look at the volume of a liver cell, right? So a reference I saw said that a liver cell is roughly cubic as it is packed in, within the organ, right? The way it's surrounded, they're not spherical. They're packed in a roughly cubic structure that are about 25 micrometers on a side, right? So the volume of that liver cell is going to be 25 micrometers cubed, or about 15,625 cubic micrometers, right? So that's the volume of a cell. So if we just to get an estimate of relative volume, so if we take the volume of our cell divided by the volume of our mitochondrion, right, that's going to be 15,625 cubic centimeters divided by our 0.4418 cubic centimeters. And so we get a relative volume of about 35,368, right, when you do the math. So a cell has about 35,000 times as much volume as a mitochondrion, right? And so, that is, a cell is volume-wise about 35,000 times as big as a mitochondrion. Okay, so, right, we've got our volumes we calculated. So another fun fact is that cells sometimes have 1,000 or more mitochondria. A large oocyte, right, an egg cell, might have 10,000 mitochondria, right? So if we take 1,000 mitochondria per cell, right, and we know that there's 0.4418, I can do this, right, cubic micrometers per mitochondrion. That says that the mitochondrion then are taking up 441.8 cubic micrometers of that cell, right? If we make it 2,000 mitochondria per cell times our same number, we get 883.6. That was really badly written, right? 3.6 cubic micrometers per cell. So if we go the other way, right, and this is really the question that my student asked me in the first place, right? What fraction of the volume of a cell is occupied by mitochondria? So if we take that, right, 441.8 cubic micrometers, divide that by the 15,625 cubic micrometers, right, times 100%, right, 100%, right, to make it a percentage, we end up with approximately 8.3% of the cell's volume is mitochondria. If we're talking about 1,000 mitochondria per cell, right? If we go to the 2,000 mitochondria per cell, right? At 2,000 mitochondria per cell, it would be double that. Would be 16.6% of the cell's volume is composed of mitochondria, right? And that's with a back of the envelope calculation here, right? And I've seen estimates of, that 40% of the cytoplasmic volume can be mitochondria in some cells. So 16% of the overall volume, I guess that's excluding the nucleus and the endoplasmic reticulum, that 40% number seems reasonable. So somewhere between 8 and 16% of the cell's volume might be mitochondria, which I find an impressive number. That's kind of cool. Okay, so let's start again with our volume of the mitochondrion, which is 0.4418 cubic micrometers, right? And so let's convert that to liters, right? Well, we know that there are 10 to the 6th micrometers. Okay, so there are 10 to the 6th micrometers in a meter, and there are 100 centimeters in a meter, right? And so, right, if we cube both of those numbers, that would convert our cubic micrometers into cubic centimeters. But then we know that 1 liter is 1,000 cubic centimeters. And so we end up with a number like 4.418 times 10 to the minus 16 liters, right? So the volume of one mitochondrion is about 4 times 10 to the minus 16 liters. Okay? Now, this is the calculation we did when I was in graduate school. So if the pH, all right, we take sort of an average physiological pH is 7.4, that means the concentration of hydrogen ions is 10 to the minus 7.4, which is about 3.98 times 10 to the minus 8th molar, right? Hydrogen ions per liter, right? So if we take that 3.98 times 10 to the minus 8th moles of hydrogen ions per liter, right, times the 4.418 times 10 to the minus 16th liters, you get 1.759 approximately times 10 to the minus 23rd moles of hydrogen ions, right, in a mitochondrion, right? So if we take that 1.759 times 10 to the minus 23rd moles of hydrogen ions, right, times Avogadro's number, 6.022 times 10 to the 23rd particles per mole, right, you end up with 10.59 hydrogen ions. That is, yes, free in a mitochondrion is somewhere between 10 and 11 hydrogen ions. So when we talk about electron transport and creating the chemiosmotic gradient, pumping hydrogen ions out of the matrix, right? We're talking pumping hydrogen ions out of a space that has between 10 and 11 hydrogen ions total in that space, right? Not 10 millimoles or 10 micromoles or even 10 nanomoles. Between 10 and 11 free hydrogen ions in a mitochondrion. That is a cool number. Okay, if we look at some other molecules associated with mitochondrion, right? Acetyl-CoA, for example. Acetyl-CoA, I found an estimate that said acetyl-CoA concentrations are about 120 micromolar, right, in a mitochondrion. So that's 120 micromoles per liter, right? 1 mole is 1,000,000 micromoles, right? And if we multiply that times our 4.418 times 10 to the minus 16th liters per mitochondrion, then we find out, and then multiply that, right? That would give us moles, 6.022 times 10 to the 23rd per mole, we end up with about 31,926 molecules of acetyl-CoA per mitochondrion, right? So again, we're down to a number, it's a much larger number, but we're down to a number where we can count how much acetyl-CoA is in a mitochondrion. Kind of cool, right? If we can consider a different molecule, right? NAD, right? The molecule NAD and its reduced form NADH, right? In cells, one estimate I saw is that it's about 0.3 millimolar total in cells. But that of that 0.3 millimolar, approximately 80% of the cell's NAD and NADH is bound, right? And they did that by fluorescence, right? Bound to some protein, right? So it makes us think differently about NAD and NADH. We think of them as free, but right, as a coenzyme that acts more like a co-substrate, but at any moment, 80% of the NAD and NADH is bound to something. But also, about 70% of the cell's supply of NAD and NADH is in the mitochondria. Right? And so, right, here we go. Right? Looking at different cells, it looks like, a decent estimate is that there's about 200 attomoles of NAD, NADH, right, in an average cell, right? 200 attomoles. And atta, if we don't remember, is the prefix for 10 to the minus 18th, right? So there's 200 times 10 to the minus 18th attomoles in a cell. And if we go back and convert our volume of a cell from cubic micrometers to liters, you get, that's times 10 to the minus 18th moles, right? That 10 to the minus 18th was our conversion. You get about one cell is 1.5625 times 10 to the minus 11th liters. And so you get a concentration of about 1.28 times 10 to the minus 5 molar, or about 12.8 micromolar, right? So in an average cell, there's about 12.8 micromolar forms of NAD and NADH. Now, if we take that same 200 times 10 to the minus 18th moles of NADH, well, I guess that's NAD and NADH combination, right, times 0.7, right, 70% in the mitochondrion, right, that gives us about 1.4 times 10 to the minus 16 moles distributed in mitochondria. If we take that 1.4 times 10 to the minus 16 moles and divide that by our total volume of mitochondria, right? Our total volume of mitochondria, that would be 4.418 times 10 to the minus 16th liters. You end up with about, doing it that way, you end up with 0.0.317 molar, right? So 0.3 molar concentration of NAD in the mitochondrion. That's quite a high concentration, right? That's a lot of NAD, right? And last, I wanted to just mention something that I found interesting about mitochondrial NAD. I'm sorry, not mitochondrial NAD, mitochondrial DNA. Little dyslexia there for a second, right? Mitochondrial DNA. Mitochondria have two circular chromosomes, which I found interesting, that are very much like the chromosomes of bacteria, right? Circular pieces of DNA. And those two circular chromosomes have a total of 37 genes total, right? And so, right, two of those are components, are subunits of the F0 portion, the membrane-bound portion of the ATP synthase. Three are subunits of cytochrome C oxidase, that is complex 4 of the electron transport chain. One is for the cytochrome B component of complex 3 of the electron transport chain. 7 are subunits of the NADH coenzyme Q reductase, that is complex 1 of the electron transport chain. One is called humanin, which seems to be some kind of protective protein. 22 are for transfer RNAs, right? Transfer RNAs. And since we only have 20 amino acids, it's interesting that there's 2 for leucine and 2 for serine. The other amino acids only have 1 each. And then 2 genes for the ribosomal subunits. Right? Now, what it doesn't have is the genes to encode its own DNA polymerase and RNA polymerase. So the DNA polymerase and RNA polymerase for mitochondria are produced by nuclear genes and then transported into the mitochondrion, as are the rest of the components for the other things there. So